Hey there, in this video I'm going to walk you through the DNA replication whiteboard activity. So if you haven't done so already, make sure you have grabbed all of your supplies. This video is going to look just slightly different than what you are going to do in this activity because you are going to use your markers to actually sketch the DNA replication model onto your whiteboard, whereas I am going to use our DNA structure model components to help illustrate in this video, just because it's going to be a little bit easier, but in the end all the same information and it should look the exact same. So make sure that you are reading the information as you are drawing and labeling. All of the information for this activity is in the reading. Okay, so we're going to start at the beginning. If you have not drawn out this image onto your whiteboard, please pause the video and do that now. So we know that DNA replication is the process of making an identical copy of DNA. This process occurs in the nucleus during the S phase of interphase. DNA replication ensures that each new cell made will have exactly the same DNA as the original cell. Recall that DNA replication occurs before both types of cell division, mitosis and meiosis. So we're going to draw a strand of DNA as it's about to undergo replication. Use the image below to guide you. So again, I am using my model. You have drawn this onto your whiteboard. So step 1, unzip the DNA. The enzyme helicase unzips the DNA into 2 strands. The opening is called the origin of replication. Technically, there are several origins that will be unzipped at one time. We only have one origin represented. There's only so much space on our whiteboards. But remember, DNA is a very, very long and large molecule, so you do technically have several origins being unzipped at the same time. So we're going to label the first step in DNA replication for our lab station. Use the image on the first page to help us. So we are going to label our replication fork. So the replication fork is right here. So replication fork. So remember, that is your origin of replication. So that is where our 2 nitrogen bases, our base pairs, have been split open. The replication fork is created by the enzyme helicase. So helicase is responsible for unzipping the 2 strands and creating that replication fork. We also have free floating nucleotides just hanging out in the cytoplasm, or really, it would be in the nucleus, because we said that's where DNA lives. So hanging out in the nucleus are free floating nucleotides right here. All right, so that is step 1. Flip my page over. Step 2, complementary nitrogen bases are added according to our base pair rule. So we are going to use a second enzyme, DNA polymerase, which is going to add complementary nucleotides to our template strands. Recall, A's pair with T's, C's pair with G's. DNA polymerase only adds nucleotides to the free 3' end of the template strand. This forms new DNA strands in the 5' to 3' direction only. So just like you read from left to right, DNA polymerase also reads left to right. We just say 5' to 3'. One of the strands is the leading strand. The leading strand is made towards the replication fork and is made continuously. So this, move some stuff out of the way, is our leading strand. So it would be 5 prime. The other strand is the lagging strand, and it is synthesized away from the replication fork and is made discontinuously. So this is our lagging strand. So this would be our 3 prime end. The replication of this strand is accomplished in short pieces called Okazaki fragments. The fragments of DNA are replicated and then stitched together by another enzyme. So we are going to label our leading strand, our lagging strand, and our 2 enzymes that are being used here, DNA polymerase. So we have 2 enzymes of DNA polymerase. One is replicating the leading strand continuously, while the other one is replicating the lagging strand discontinuously. This is important because, again, remember, we can only read 5 prime to 3 prime. So essentially what is happening is as this DNA polymerase is moving, it's following helicase. So helicase is coming in and it's unzipping these bonds, and then your DNA polymerase is coming in afterwards and is adding in the correct nucleotide. So, for example, there we go. We have G and C here. Pretty sure this is a T, so I'm going to add an A. And then there's another enzyme that comes in and binds together our phosphates and our sugars. We don't necessarily care about that enzyme. I can't get this. There it goes. Okay. So all my DNA polymerase is doing is adding in the complementary base to create that new strand of DNA. This DNA polymerase is doing the exact same thing. It's just having to do it discontinuously because helicase is moving this way, and as it's unzipping, DNA polymerase is having to move from the 5' end to the 3' end. So it'll do this small chunk, and then it'll go back, and it'll do the next chunk. So it's discontinuous. It's a lagging strand. But it's still just DNA polymerase. There's just 2 of them. So step 3, 2 identical DNA molecules are formed. So eventually helicase will come through and will break, right, all of those bonds. So helicase has moved through. And then we will add in our complementary base, which is done by DNA polymerase. So DNA polymerase moves through, adds in our complementary base. So this is why I'm having you draw it. But it's okay, I can get there. This one, oh, I left my phosphate. There we go. And remember, all of these nucleotides, they're just free floating in the nucleus, waiting to be used in DNA replication. So here's this one. Add that one in, and then I need another. A, T, C, and G, following my base pair rule. Oh, lift off my covalent bond. There we go. So I then have a whole new double helix structure, right, following helicase, and this structure would do the same thing, except it would be discontinuous. So what that means here, we would have this side, where's my bond? My G and my C are combining. I have A, oops, ripped the whole thing off, paired with a T. So DNA polymerase moves, it adds these two new base pairs, and then it's got to go back and it's got to do the next fragment. So it's a lagging strand because instead of the replication happening continuously, it happens in small chunks. So at the end of the day, we still end up with, right, our correct structure. We still end up with a whole new strand of DNA. Oops. It's just having to occur in small fragments, Okazaki fragments technically, named for the guy that discovered this. So polymerase is done here. Now it's going to come back, right, and it's going to do the last fragment, bind these bonds, and then the last one. Oops, almost messed up there. That would have been really bad. Yay! So now at the end of DNA replication, I have two new identical daughter strands of DNA. They are identical. I want them to be identical. I don't want any mistakes, right? DNA mutations. So we say that DNA replication is semi-conservative because each of the original template strands are used to build the new strands. So these outside strands, right, they were part of our original DNA molecule, and so we only had to build the other half. So DNA replication is semi-conservative. So there we go. I now have two new strands of DNA. They are identical, which is awesome because that's what I want. So hopefully this video helps at least a little bit in understanding the parts of DNA replication. There's a lot more that goes on, but we are really only focusing on kind of the major points. Our enzyme helicase, right, which breaks the hydrogen bonds, and then DNA polymerase enzyme, which is responsible for adding in the new nucleotides to both the leading and the lagging strand. Okay? Don't forget to complete the DNA replication activity sheet. This is yours to keep in your notebook for your mastery check. But still, if you have any questions or concerns as you're doing this activity, just let me know.